So we will now call upon our second explorer. She's Kavita. Kavita did her undergraduate education in physics, chemistry and mathematics from St. Joseph College, Bangalore, and went on to obtain a PhD in developmental biology from the National University of Singapore. She began working on a worm called C. elegans at Massachusetts General Hospital during her postdoctoral studies. So she headed her lab at IISER Mohali for close to 8 years, after which she moved to Indian Institute of Science, Bangalore a few months back. Kavita continues to be fascinated by the mind of the worm and the mind of the criminals, as she just told us that she loves to watch crime movies. And she loves to watch cooking shows as well. So we will see today what story she will be cooking up today. Also, a little fun fact about Kavita is, so if you thought that you can eat a lot of ice creams, she just told us that she ate 50 scoops of ice cream in a single sitting. So yes, let's welcome our second explorer, Kavita.
And first I want to thank India Alliance for giving me this opportunity, and let's see, so what I'm going to talk to you about is how neurons talk to each other. Essentially, that's something that I've been interested in for many years. A little bit about me. So when I was in 7th, 8th standard, I really hated maths. I was very bad at it. I just wasn't getting it. But by the time I reached 10th, for some reason I managed to understand it, and it became like a puzzle that I needed to solve. And I really became intrigued by maths, which is why I did my undergraduate in maths. But at the same time, I was also starting to really begin to understand biological concepts, and also became very interested in biology. And to me, they're both very linked. Essentially, they're like a crime, that you're actually trying to find out who is doing what. And that's one of the reasons why I find both biology and math, I haven't been doing it in many years, very intriguing. And today I'm going to tell you something that might be a little bit like solving, like being a detective and solving what some protein does in our body. Okay. So, what I'm going to tell you today is how neurons talk to each other, and we're going to do this from a different perspective, from the perspective of a worm. So all of you know that your brain is very important, right? Whether you're thinking, you're walking, you're talking, everything requires functioning in the brain. And essentially, the brain actually senses out what is outside, what is in our surroundings, and then tells our body to act accordingly. So if you wanted to eat something, for instance, your brain actually says, okay, this is something, you're lifting it up, putting it in your mouth, and from the sensations that are there in our mouth, the muscles of our mouth are actually activated, and they then allow for chewing of the food. So essentially, when you think about it, the neurons are doing everything that we do. And these neurons are constantly talking to other neurons. Each neuron in our brain is making connections to many other neurons. So there's always so much chatter that's happening in our brain, and that's what's allowing us to do everything from sitting here to getting up, walking, talking, everything requires that. So just to give you a very brief introduction into how a neuron talks to another neuron. So what we have is essentially, I don't know, all of you must have seen a schematic of a neuron, right? It's made up of a cell body followed by a long axon. And I'm not going to talk about the cell body and the axon part, but you have the axon that comes down like this. And at the tip of the axon, you have a region that allows for connection with the next neuron. Okay, so you have a current that comes down the axon, the entire length of the axon, and this then allows calcium to enter into the tip of the axon. This calcium, once it enters, causes a chemical to be released. This chemical then gets released from this neuron. The chemical then binds, so it actually interacts with a protein that sits on the surface of this other neuron. The other neuron is the neuron this one is talking to. Once that interaction happens, happens? The next neuron gets activated. And this process is done thousands and thousands and thousands of times every minute in our body, and this is what allows us to do pretty much everything that we do. Okay, so, but is this the entire story? So, I've already told you that we have this chemical, it binds this other protein, and this allows for this cell to work. But is this the entire story? Are there parts of it that we're missing? And that is indeed the case. There are also lots of molecules that are sitting both in the first neuron as well as in the second neuron. And these molecules are talking to each other and they're allowing for this process to happen smoothly. So what I'm interested in doing is understanding who these molecules are and how these molecules function, right? So it's a very straightforward question. Who are these molecules that are sitting here and how do they function? How do they allow for this smooth talking between this neuron and this neuron? Okay, so again, let's go back to the human brain. We have more than 10 billion neurons in our brain. So that is a huge number, and more than 10 trillion synapses in brain. Actually, I'm sorry, this is actually 100 billion neurons that are there in our brain, which is a huge, just think about it, the number of zeros that has. That's a huge number of neurons. And each of these neurons makes so many connections with other neurons, making more than 10 trillion connections. So just think about it. How do you start studying this process? How do I look into one of your brains and say this neuron is making a connection here, so now let me start studying who are the molecules, what are the proteins that are there in both the neurons, right? It's very difficult. So instead of that, we look at a very simple and elegant model, which is called Synorhabdatus elegans, or the worm. Okay? So this is the worm. And if any of you want to go and see worms, they're there under that microscope. You can go wherever you get a chance, you can just go. There are 3 plates there, you can just move around, see whether you can actually see the worms. Okay? The worm, on the other hand, has 302 neurons. The number is known, it's very simple, very straightforward, and not so many synapses, just 7,000 synapses. However, what is very interesting is that humans have 25,000 genes. So you and I have 25,000 genes that code for all our proteins in these 25,000 genes. C. elegans, although it has very few neurons, very few synapses, has very similar number of genes. It has 20,000 genes. So now we can start thinking, okay, are the genes similar? And many of the genes are similar. The genes that are required to make the chemical that is sent out from the neuron is similar. The genes that are required to make the protein that the chemical senses are similar. All of that is similar, making it a very good system, right? So now I can remove those proteins from this small model organism and see what happens. And then I can go and see, okay, does the same thing happen in vertebrates, and then maybe the same thing happens in humans. And then we can start thinking about, can we now target these molecules? Okay, I'm not going to go to more than worms, I'm just going to tell you about worms. So just to give you a little bit of an insight, that's the microscope that you'll be looking at. You can look at the worms, and these are what the worms look like. You're going to see different stages. You're going to see these eggs. There aren't many middle stages. You will only see old ones and very small ones on the plate. And essentially, these worms just eat bacteria. So it's very simple. You just take a plate with agar on it. You put some bacteria, and the worms are happily grown. This is just a life cycle, and you're going to see some of these stages in the microscope. But another very interesting thing about the worm is that if I wanted to see where a protein is in the worm, I just add GFP to the protein. GFP is a fluorescence protein called the green fluorescence protein, and I look under the microscope, and I'll very nicely be able to see all the neurons in green color. I'll be able to see the axons in green color, and I'll be able to see everywhere where that protein is. So this makes it very easy. All I have to do is put GFP with any protein and look under the microscope, I see everything I want to. Right? So it's very straightforward. So now the question comes, the question that I'm interested in asking is, who are the proteins that are sitting in the first neuron or the second neuron, and how are they actually functioning? So in order to simplify the system even more, see, now I've already simplified it from the human brain to a small organism, right? Now I'm going to simplify it even more,
And what we look at here is actually how the neuron talks to the muscle. I just told you initially that everything we do requires some sort of muscle action, right? If you want to walk, your foot muscles need to be activated. They have to get some input from a neuron that says, okay, now you walk. So what I have even more simplified it, I am now looking at just one set of neurons that talk to the muscle. These neurons send out a chemical which is called acetylcholine. And our neurons also send out the same chemical called acetylcholine, which then talks to the muscle. So essentially we have the same system. The muscles in my hand are connected by neurons that send out the same chemical as the muscles that are there in the body of this one. So the question that I want to ask is how is acetylcholine sent out and how is this muscle staying how it is, right? So a very simple way of looking at this is actually using a drug. So now you think about the fact that you have a neuron, sends out acetylcholine and activates the muscle. If you have too much of acetylcholine, what is going to happen? The muscle is going to be very activated, right? More activation of the muscle. And if you have too much muscle activation, the muscle will then get tired. It cannot keep contracting. The muscle, when you're walking, actually contracts. And if you have too much acetylcholine, the muscle keeps contracting and then you will no longer be able to walk. So we use exactly the same principle here, where we cause too much acetylcholine. So we use a drug which prevents the breaking down of acetylcholine. So when we put that drug, the acetylcholine is not broken down and you have too much acetylcholine. Now what happens is the muscle contracts and the worm paralyzes. And this is just a video that's where I'm going to show you how the worm paralyzes. So this is just a normal worm that's not paralyzing. And here you can see that once you add this drug, the worm is not able to move the same way, right? It's trying to move, it's not able to move, and after some time, after 2 hours, it will start paralyzing. So just to show you here, you can just plot at 0 time point, no worms paralyzed, and by 1 or 2 hours, all the worms paralyzed. So now I'm going to break down one very small story into 4 different topics and just tell you a little bit about each. And I'm going to put this in the form of a puzzle. I started off saying that I'm very interested in, actually finding out what happens. Right? So let's think about this in the form of a puzzle. The first thing I want to find out is what happens when I remove a protein. So in this, putting a drug and seeing what happens to paralysis, I just want to find out what happens when I remove different proteins. So what we did is we removed different proteins, one at a time, from this worm, and then see what happens. Do they paralyze faster or do they paralyze slower? Okay, and I'm just going to show you one protein in here. This is the control that paralyzes at some given speed. And you can see this one protein does not paralyze at all. Right? So it's just, it really does not, most of the worms are just not paralyzing. So now I come up and say, okay, now I have a phenotype. I can now think about what is causing this phenotype. Right? So I said if you have too much muscle contraction, that gives rise to paralysis. So maybe in this case, there is no muscle contraction. Muscle is not contracting. It's very little muscle contraction. So paralysis is very little. Right? So what could be the reason that you have less muscle contraction? One possible thing, you have less of the chemical that's required to contract the muscle, right? If you have very little of the chemical that's required to contract the muscle, there will be less muscle contraction. Very straightforward. So one can think that there's less of the There's less of the chemical that's required to contract the muscle. The second possibility is there is less of the receptor that is, there's less of the postsynaptic receptor that the chemical binds to. So these are the questions that we ask, whether is there less chemical, less receptor. But let me just take you through this as a puzzle, right? So the first question is, where does this protein actually function? So this will then tell us, is it less chemical or less receptor? Because if it functions in the neuron, it might be less chemical. If it functions at the muscle, it might be less receptor. So what do? We put back this protein into the neuron, and we put back this protein into the muscle. And we put it back very specifically, because this is a very small organism where you can do lots of different things. We put it back specifically into the neuron or specifically into the muscle. And you can see that in this mutation, the worms don't paralyze at all. You put it back into neurons, they're not paralyzing. But you put it back into the muscle, they just become like the control wild type animals, they paralyze. So now we say, okay, we know what happens when the protein is removed. It doesn't paralyze, and we put back the protein into the muscle, and we get back the paralysis. So now we know this protein is working in the muscle. Now the next question is, what does it do? I already talked a little bit about it. It's either going to cause less of the chemical secretion, which is unlikely because it's present in the muscle, so it must be affecting something in the muscle, or it's going to cause less of this protein that's sitting in the muscle. And again, I just want to reiterate, same chemical is present in you and me, same protein is present in you and me. Although I'm looking at this small organism, the same things are present in you and me, right? So then we look at the protein. I'm sorry you can't see this very well, but when we look at the protein that's present in the muscle, there is very little of this yellow stuff that you see here. Very little of that present in the mutant. So this now tells us there's very little of this protein sitting in the muscle, right? So now we've also solved what does a protein do. Now the next question we asked is, what is the significance? How could it affect this worm? This protein not being there, how does it actually affect the worm? Does it actually have any significance of not being there? And I told you initially that everything is current. So the neuron, there is a current that passes through the axon of the neuron that allows for the chemical to be released. This then binds to the protein that sits here and activates this muscle. So this current is transferred here. So if I wanted to actually look at the current, I could just put an electrode onto this muscle and I can actually quantitate how much current is there. So now you think about it, if you have very little protein that's sitting on the muscle, the current could also become less, right? Because now the muscle is not being activated. There is not much activity in the muscle, so there will be much less current. And that is exactly what happens compared to wild type, which has normal current. There is very little current in these mutants. So now we have a story where we know that this protein is sitting in the muscle. It is allowing to keep these proteins together so that the muscle can function normally. So we've essentially completed this whole puzzle essentially, where we've also got what the significance of this protein is. However, is this the end, or is there something more that we need to do? Obviously, the next question would be, how does this protein work in you and me? How does it work in vertebrates? How does it work in humans? That is a very important question, right? Because if it is actually required for the same process, then you can imagine that in a case where you have too much muscle activation, like epilepsy, you can imagine that you want to see how this protein actually functions. Because I said the same proteins are present in you and me. So how can we actually look at what is happening in humans? However, what I showed you earlier was a very nice, very systematic puzzle that's going from point A to point B to point C. But in real life, that is not necessarily the case. You might have some things that don't fit as nicely, and it doesn't matter. Because at the end of the day, you're looking at it from a biological system point of view. So you're looking at a biological system and what it is telling you. So you do not worry about everything fitting very neatly in a puzzle, but you do want to find out more of the puzzle, and you want to explore more to see what this is doing and whether we can find relevance between this organism and vertebrates and humans. Okay, with that I just want to give a quick acknowledgement where I thank a lot of people from my lab. This is my lab in ISER Mohali, taken towards early 2019. And I also want to thank India Alliance, DBT, ISER Mohali and IISc for funding. And with that, I want to take questions. You can ask me any questions about anything, including eating 50 scoops of ice cream. So
let's thank Kavita for the wonderful talk.
Puzzle. The first thing I want to find out is what happens when I remove a protein. So in this, putting a drug and seeing what happens to paralysis, I just want to find out what happens when I remove different proteins. So what we did is we removed different proteins, one at a time, from this worm, and then see what happens. Do they paralyze faster or do they paralyze slower? Okay, and I'm just going to show you one protein in here. This is the control that paralyzes at some given speed. And you can see this one protein does not paralyze at all. Right? So it's just, it really does not, most of the worms are just not paralyzing. So now I come up and say, okay, now I have a phenotype. I can now think about what is causing this phenotype. Right? So I said if you have too much muscle contraction, that gives rise to paralysis. So maybe in this case, there is no muscle contraction. Muscle is not contracting. It's very little muscle contraction. So paralysis is very little. Right? So what could be the reason that you have less muscle contraction? One possible thing, you have less of the chemical that's required to contract the muscle. Right? If you have very little of the chemical that's required to contract the muscle, there will be less muscle contraction. Very straightforward. So one can think that there's less of the chemical. There's less of the chemical that's required to contract the muscle. The second possibility is there is less of the receptor that is, there's less of the postsynaptic receptor that the chemical binds to. So these are the questions that we ask, whether is there less chemical, less receptor. But let me just take you through this as a puzzle, right? So the first question is, where does this protein actually function? So this will then tell us, is it less chemical or less receptor? Because if it functions in the neuron, it might be less chemical. If it functions in the muscle, it might be less receptor. So what do? We put back this protein into the neuron and we put back this protein into the muscle. And we put it back very specifically, because this is a very small organism where you can do lots of different things. We put it back specifically into the neuron or specifically into the muscle. And you can see that in this mutation, the worms don't paralyze at all. You put it back into neurons, they're not paralyzing. But you put it back into the muscle, they just become like the control, wild type animals, they paralyze. So now we say, okay, we know what happens when the protein is removed, it doesn't paralyze, and we put back the protein into the muscle and we get back the paralysis. So now we know this protein is working in the muscle. Now the next question is, what does it do? I already talked a little bit about it. It's either going to cause less of the chemical secretion, which is unlikely because it's present in the muscle, so it must be affecting something in the muscle, or it's going to cause less of this protein that's sitting in the muscle. And again, I just want to reiterate, same chemical is present in you and me, same protein is present in you and me. Although I'm looking at this small organism, the same things are present in you and me, right? So then we look at the protein. I'm sorry you can't see this very well, but when we look at the protein that's present in the muscle, there is very little of this yellow stuff that you see here. Very little of that present in the mutant. So this now tells us there's very little of this protein sitting in the muscle, right? So now we've also solved what does a protein do. Now the next question we ask is, what is the significance? How could it affect this worm? This protein not being there, how does it actually affect the worm? Does it actually have any significance of not being there? And I told you initially that everything is current. So the neuron, there is a current that passes through the axon of the neuron, that allows for the chemical to be released. This then binds to the protein that sits here and activates this muscle. So this current is transferred here. So if I wanted to actually look at the current, I could just put an electrode onto this muscle and I can actually quantitate how much current is there. So now, you think about it, if you have very little protein that's sitting on the muscle, the current could also become less, right? Because now the muscle is not being activated. There is not much activity in the muscle, so there will be much less current. And that is exactly what happens compared to wild type, which has normal current. There is very little current in these mutants. So now we have a story where we know that this protein is sitting in the muscle. It is allowing to keep these proteins together so that the muscle can function normally. So we've essentially completed this whole puzzle essentially, where we've also got what the significance of this protein is. However, is this the end, or is there something more that we need to do? Obviously, the next question would be, how does this protein work in you and me? How does it work in vertebrates? How does it work in humans? That is a very important question, right? Because if it is actually required for the same process, then you can imagine that in a case where you have too much muscle activation, like epilepsy, you can imagine that you want to see how this protein actually functions. Because I said the same proteins are present in you and me. So how can we actually look at what is happening in humans? However, what I showed you earlier was a very nice, very systematic puzzle that's going from point A to point B to point C. But in real life, that is not necessarily the case. You might have some things that don't fit as nicely, and it doesn't matter. Because at the end of the day, you're looking at it from a biological system point of view. So you're looking at a biological system and what it is telling you. So you do not worry about everything fitting very neatly in a puzzle, but you do want to find out more of the puzzle and you want to explore more to see what this is doing and whether we can find relevance between this organism and vertebrates and humans. Okay, with that I just want to give a quick acknowledgement where I thank a lot of people from my lab. This is my lab in ISER Mohali, taken towards early 2019. And I also want to thank India Alliance, DBT, ISER Mohali and ISC for funding. And with that, I want to take questions. You can ask me any questions about anything, including eating 50 scoops of ice cream. So
let's thank Kavita for the wonderful talk.
So, I have a question that just clicked my mind, that you've been talking about the neurons and all that, talking between those neurons and when the messages go to our body parts. So I was just asking a question that, do you feel that, or do you, can you please, prove that, or you say that the speed of the light is I would say the thinking process or the thought process or the messages between the neurons is much faster than the light of the speed, and if you think so, that how could you justify it scientifically?
It's very fast. I'm not going to say that it's faster than the light of speed, but the way you can actually find out how fast it is the speed is by essentially doing this. What you do is you just patch onto the neuron. You put an electrode onto the neuron and that will measure current. Okay? And then you just look at, and as I showed you here, you see this huge current, right? Coming down very quickly. So you just measure how quickly that happens. So you can measure that for multiple, currents. You keep seeing current. That will give you how quickly the first neuron is talking to the second. So just by actually measuring electric current, you can actually find out the speed. Does that kind of answer your question? I don't think it's faster than the speed of light. That is wrong. There are billions of neurons. In humans, there are 100 billion neurons. Very difficult to figure out. So, and what I'm interested in is actually looking at molecules. What do these molecules do? And for that, it becomes much easier when you have a very fixed population of neurons, and you can keep knocking down molecules very easily, and then you can say, okay, this is what this molecule does. And then we can go on to higher systems to see if we can validate that.
From there, then paralysis would not be happen. So, can this model be used in humans to get back from paralysis?
So, okay, this is how it's working in CLIS. Now, let's look at something bigger. Let's look at mice. Does it work in the same way in mice? And then if it does, then can you target it? Can you somehow use some target to prevent this molecule from working and then go on into humans? And so that's something so this is essentially very fundamental research that I work on, but one hopes that this fundamental research can be taken up and used for translational research.
Okay.
Muscles distinguish between different signals. Like if my hand is, I'm opening my hand and closing my hand. How does these two things distinguish?
Okay, so I'll tell you how it happens. So I don't know if you've ever seen CLS. It moves like this. It's like a snake, and you can go and see that. Okay, so it moves like a snake. So the muscle essentially gets two neurons inputting that give it messages. One is acetylcholine. Acetylcholine is what is there in you and me. It causes muscle contraction. The other is GABA. And GABA causes muscle relaxation. Okay, so one side, so when the worm is moving like this, one side is contracted. And the other side, on the opposite, this side is contracted. So this muscle is contracted. This muscle is relaxed. So the other muscle gets GABA, when one muscle gets acetylcholine. So you can imagine that you have a different set of inputs coming onto muscle, and that allows for contraction or relaxation, or adjust the amount of contraction and relaxation. That is one way by which you can think about it, but there could be more processes that are involved that are still not known.
Like the muscles are guided by neurons, and neurons are guided by brain. So who guides the brain to send some signals?
Oh, who guides the brain? The external. That my brain is. So there are neurons in my eye. There are sensory neurons in my eye. Those are getting visual cues from here. That is telling my brain, okay, now I'm seeing these people.
Are you able to hear? I'm seeing these people, and now I, have to appropriately decide what I want to do. So if I'm giving a talk, I decide, okay, I have to go come here and give a talk.
So it's like a cycle.
Yes, exactly. It is a cycle.
I've already asked many intelligent questions. I would just like to ask which flavor the ice cream was.
Base was a sports fund where you give $5 and you get a spoon. And all the local ice cream people set up their stalls there. So all the flavors of ice cream. So all lots of different things.
Thank you.
Biologists.
Is there any way we can give them life back?
Okay, so it's known that it's not. So I don't know if you've seen the classical frog experiments where you cut off the head of the frog, but the legs are still functioning. So if you electrically spark the leg will kick. So as soon as the head is cut off, there is still activity in the muscle. There is still activity in the neurons. So maybe you can think of some way of actually keeping that activity. But then my question would be, why would you want to do that?
So basically, if we can regenerate memories from neurons, retain memories from neurons which are stored in them. So basically, take Newton. So maybe he is there, of course. So if we are able to regenerate or retain memory from its neurons, so we're able to find many things which he has not written or not explored.
Okay, I think that's a good idea, but I think there's also a lot of ethical concerns with trying to do something like that. So that needs a lot more thought before you go ahead and even try to simulate something like that.
We can certainly take more questions during our break. So let's thank Dr. Kavita for sharing.